Thank you so much, Chris, and thank you for the invitation to speak. So I will jump right into this. We just had such a wonderful panel discussion, so I'll skip lots of introduction for you all. But I am Dylan. I am a postdoc based in Johannesburg, South Africa, and I'll be presenting work from my team where we've looked at expanding the representation of African populations in human gut microbiome research. So in this project, this was an international study focused on genomic and environmental risk factors for disease in adult populations in South Africa, Ghana, Burkina Faso, and Kenya. In AWI-Gen, we enrolled over 10,000 individuals, and this involved extensive survey data, clinical measurements, and collection of blood, urine, and stool samples. As a sub-study of AWI-Gen, we enrolled 1,800 women, so all adult women ages around 40 to 80, from the 6 study sites to study the gut microbiome. And so by cross-sectionally enrolling individuals from these 4 countries, we can begin to shift the focus from microbiome research that is exclusively on populations in high-income countries where there's relatively homogeneous lifestyle, diet, and exposure, and instead work towards capturing the 83% of the world that resides in low and middle income countries where there's much more diversity of diet, lifestyle, and disease. So on the right-hand side here, you can also see the main research team for the AWI-Gen Microbiome Project, and this represents an international team of professors and postdocs from both Stanford University and Wits University in South Africa. So here are just a few photos of some of the study sites. As you can see, these sites span many different settings, from areas in West Africa, like Nanoro, Burkina Faso, and Navrongo, Ghana, that are rural, agricultural. Subsistence is primarily from small-scale farming. We see industrializing rural communities that have much greater access to electricity, to piped water, to shopping centers. We also sample from 2 informal settlements and high-density areas in Nairobi, Kenya, where there's much more limited access to clean water and sanitation. And then we sample from individuals in the Soweto township of South Africa. And so you can see here that we're capturing a very broad range of lifestyle and health diversity. So today I won't be talking about any of the scientific findings from our study. You can see them in the abstract, you can check out our paper, but instead I'll be focusing on some of the lessons that our team learned about how to conduct ethical microbiome research in underrepresented and marginalized populations. And so we've already heard a lot of wonderful talks today about some of the open questions and considerations in this research, but I'll try to provide some specific examples of what has worked well for us in this collaboration. And so I'll start with a discussion of how we've navigated this equitable collaboration between a high-income country institution and institutions in LMICs. I'll start by sharing a quote from a very dear friend of mine from Ghana, who works between the US and South Africa, and he's currently based at one of our field sites in South Africa. And so talking to him about this presentation last week and soliciting some of his ideas, he shared this quote with me. So he says that US institutions use African institutions to collect data, and then they go. In a meaningful collaboration, you collect the data together, not from your computer at Stanford. And when it comes to data analysis, you work through it together and you decide on the papers together. That is what makes a real collaboration. So one hugely important element of this for us in AWI-Gen was leveraging existing research infrastructure rather than jumping into these new settings as American researchers and dictating the terms of a project for which we did not understand the cultural context. So in AWI-Gen, we partner with several health and demographic surveillance centers that provide research infrastructure that enables us to connect with and work with local researchers and engage them in the project from the conception all the way through preparing manuscripts. A second element that we've prioritized is thinking about the leadership and advancement of trainees from both sides of our partnership. One really amazing opportunity has been the NIH Fogarty Global Health Equity Scholars Program, which funds trainees from both the US and abroad to conduct research exchange. And this has funded both a trainee from Wits, Voke, here in the top left, and me as a trainee from Stanford to participate in a research exchange where we travel and receive mentorship from the other institution. So now I've been based in Johannesburg for almost a year at this point and have been wrapping up my fellowship. We also prioritize first and corresponding authorship representing trainees and faculty from both institutions, and inclusion of authors from every study site, as you can see in our 3 papers here. And importantly, this authorship inclusion is not something that is perfunctory, but related to specific contributions that have been carved out and dedicated to researchers from across the field sites. Now, I'll change gears a little bit to talk about ethical engagement with study participants. And again, I'll start by sharing a quote from another friend of mine who was a doctor in Zimbabwe and also now works at one of the field stations in South Africa. So talking to her about this, she said that people in these studies want to benefit from the research that's being done. They want to see implementation and feedback. And if you don't come back and give feedback and explain it, then there is no point in their participation. So one of the most critical elements of participant engagement is engaging the community before a project begins. So in AWI-Gen, for each study site, researchers engaged with the community members to discuss feedback and concerns related to the study. And this was adapted to the local context. So depending on the site, this ranged from meeting with chiefs, with tribal leaders, village elders, larger community advisory groups, and their feedback shaped the selection of the target population, the focus of the research questions on issues of local importance, rather than what we heard about in the panel discussion where researchers tried to market microbiomes of these populations for health improvement in the global north, and then also shaped consideration of cultural sensitivity surrounding sample collection. But also very importantly, when thinking about community engagement, it is so critical to have follow-through and return to the community after the study. So many of the study communities that we work with in AWI-Gen have become very familiar with researchers from WITS or from the US or from Europe coming in, conducting a nice advisory group session, taking huge amounts of blood samples especially, and never returning results. So over the past few months, we've been working on a framework for returning microbiome results to participants. And here you can see the translated information sheets that we've been giving back to participants. So this includes 1 page of general information on our study findings, and then an individualized report on their microbiome data. And so returning these forms is a huge undertaking in and of itself. A few weeks ago, I traveled with my team to Agincourt to interview and hire these 2 wonderful field workers, Carol and Miriam, who are pictured here on the left. And we spent several days teaching and learning about concepts in microbiology and microbiome research. You can see on the right that we bought these origami foldable microscopes so that they and our participants can actually see microorganisms in real time. We did some extensive office prep from mapping out in this study site where the 500 participants live across this 400 square kilometer catchment area. We prepared all of the individual forms to be returned. And then we did a lot of driving through the villages to find participants. So you can imagine the time and effort it takes to track down 500 individuals across this huge rural area where people may not be home, they may be out at work, they may be too busy to speak, and then taking the time at each house to knock on their door, introduce ourselves, ask to meet, and then sit outside like you can see on the right. That's Carol and Miriam again, often sitting for 20 or 30 minutes going through one-on-one with participants, their results and all of their questions. And so as you see, this represents hundreds of hours of meeting with participants, not including drive time and office time, and then thousands of dollars worth of resources as well. So this effort is still ongoing. We just got started and we're just in 1 site right now, not all 6, but we've learned so much so far. First, we found that community engagement is something that should be an absolute requirement and not an afterthought. It's something that should be planned for and budgeted for at the start of a study. We saw that participants really want their data and they have a fundamental right to their own information. And we found that participants are happy to learn about their data even if it's not clinically actionable, the same way that a blood pressure result or an HIV test might be. Further, we found that even among the participants that we work with, where a good portion of the population is not literate or numerate, everyone can understand concepts in microbiology if they're given the time and the space to learn and ask questions. And so I recognize that most of the microbiome folks on this call may not be doing studies in LMICs or even doing primary data collection, but we can learn a lot from the talks today. Like Katie said, we can hold ourselves to a higher standard when reviewing papers, not just asking for methods about collection kits, but how communities were engaged. We can recognize the value of community engaged work. As you saw, community engagement in one site cost over $10,000 and hundreds of hours of work. So we need to value that appropriately when we're reviewing. And finally, we can continue to learn and reevaluate our biases. Many of us are guilty of infantilizing study participants and collaborators, asking, why would a participant in rural South Africa care about their microbiome results? Why would a field worker in a rural area want to be an author on a paper? But we can continue to do better here and to learn and grow and challenge our own expectations. So with that, I'll conclude by leaving you with a homework assignment. The first one is optional, which is to check out our preprint for the scientific results. It's linked here. The second is not optional, which is to read these two articles, which for me at least were very thought-provoking in thinking about how to do research in populations around the world. Finally, I'd like to thank you all so much for your attention. And of course, thank the hundreds of people involved in this project, the thousands of participants, the staff and field workers at all the study sites, and the amazing scientists who have hosted me at WITS for the past year. Thank you all so much for your attention, and I am looking forward to your questions. Thank you.